Thank you, Professor Power, for the nice introduction, ladies and gentlemen. And I thank you very much, not only for the introduction, but for the invitation to speak here at the occasion of the LSD Symposium. I appreciate that very much because I became acquainted with Dr. Hofmann years ago, and so I liked very much the occasion to see him again. And I like to draw your attention to the fact that natural products, especially from plants, are either poisons or medicines, and that there is a close relationship between poison and medicine. And I will try within the next few minutes to show you some examples how these close relationships developed during history. And I will talk a little bit about examples of pure plant constituents, then the modification of plant substances during history, and the use of plant substances as models for medicinal products, and then a short thought on mixtures from plants we use still as herbal medicinal products for therapeutic purposes. We don't know how everything started. Our ancestors probably were looking for plants for nutrition purposes, and they found out that basically every plant can be eaten. Some plants only once, if you think of poisonous plants, and they probably have found out very quickly that there are very poisonous plants, and probably it took longer time to find out that several plants have healing properties as well. If we jump through history, we find the earliest written records in China more than 5,000 years ago, and then in India, think of the Ayurvedic system. And then, as an example, I took the famous Papyrus Ebers, written in Egypt 3,500 years ago. And there already squirrel is mentioned and described as medicinal plants. I hope you can read this transcription. If not, we jump further in history, and we come to the ancient Greeks, and they used the term Pharmakon already in a double sense. It was the name for poison and at the same time for antidote, for remedy or medicine. And this ambivalence goes through the centuries up to now. Several substances, dependent on the dose which are used, atropine as example, dosis facit venenum, we heard that already. Paracelsus said this already. It depends on the doses whether atropine is a poison or it can be used as medicine. It depends on use or misuse of a substance. Think of morphine or LSD. It can be used as medicine or as poison for the single individual. And then structure modification makes a substance from medicine to a poison, probably. The ancient Greeks had a good knowledge of poisons already. We heard that in the morning session already again. And I remind you of Socrates. He died of poison hemlock, which was a legal device for execution at the ancient Greeks' time. So the term pharmakon is derived from the antique Greek culture. And if we jump again, we find several physicians, famous physicians, you know all the names. I took out one example, Dioscorides. He lived in the 1st century after Christ, and he described about 600 plants already. As an example, I took squill again, and this drawing comes from the so-called Wiener Dioscorides, written 512 in Constantinople. And keep in mind this drawing of the plant. In the books of Dioscorides, there are already mentioned alexipharmaca, helpful pharmaca. So if you became poisoned, you needed an antidote, an alexipharmacon, and theriaca are described in this book already. So this book was written 500, this handwritten book stems from 512. We jump 1 millennium and come to Basel, where a famous book was edited, written by Leonhard Fuchs in 1543. And again, more than 500 plants were described by Leonhard Fuchs. He had a good knowledge of medicinal plants. And I took the picture of squill again, and you don't see so much a difference, although 1,000 years were in between the two pictures from Dioscorides to Leonhard Fuchs. And we jump again 200 years, and we come to 1785. And this man is William Withering. He was physician at the General Hospital in Birmingham. And he got a recipe from a lady who took several crude plant drugs and used it against dropsy. And it was he found out that in this mixture that the leaves of Digitalis purpurea, of Digitalis, were the active principles. So he tried 10 years with his patients. He made human trials to find out the exact dosage, and he used water extracts from the leaves of Digitalis purpurea. So the poisonous plant can be a medicine if you use the right dose. This was the book of William Withering, and it's important. But the first real big progress came up in the 19th century when natural sciences contributed to medicine. And here are a few dates only that pure plant constituents were isolated. A very important date in my eyes is the isolation of morphine, and you see a lot of other strongly active plant substances were isolated in the 19th century. For example, 1833 atropine, 1860 cocaine. We will come back to this briefly, and digitoxin. And chemistry developed very rapidly, and it was possible to undertake pharmacological investigations with the pure plant constituents. This is the example, 1805, the German pharmacist was able to isolate morphine from opium for the first time. And just a few pictures, opium poppy in a field as it is cultivated, and the plant, and the ripening capsule is used for producing opium. And up to date, the alkaloids are produced from opium or from poppy straw, morphine, codeine, and noscapine. So this was 1805. Morphine as a new substance and as a pure substance. Another example is cocaine. And in Europe, it was very well known that South Americans were used to chew coca leaves, and it was reported in the 16th century already. But only in 1860, the first amounts of coca leaves suitable for analysis were brought to Europe. And immediately, Niemann in Germany succeeded to isolate pure cocaine. And this interesting substance was used for several trials. One example is given here. Sigmund Freud in Vienna, well known, made trials with his patients and tried to use cocaine against morphinism. But these trials were not successful. He came from the frying pan into the fire and produced morphococainists. So you see a new substance, and he tried to use it as a medicine, but he did not succeed. But at the same time, the discussions among the medical doctors in Vienna led to the discovery of cocaine as local anesthetic by Karl Koller. And since that time, it is used as a very important local anesthetic in ophthalmology and so on. So new substance, and it took some time to find out, is it useful as medicine, or can it be used for other purposes? And people tried it as a drug against nausea, vomiting, and other diseases, and even art was inspired, as you can see by this book printed in Berlin, 1900, modern art inspired by Mama Coca. This is Mama Coca with a twig of the coca plant, and these are the coca leaves as they are chewed in fresh state in South America, as you know. So there was even produced a sparkling wine in Vienna containing cocaine. And it was used against nervous diseases, against stomach diseases, and generally against every weakness even. So it took again some time until people realized that cocaine cannot be used as, for everything and as medicine. It turns out to be a poison, if you like this expression here. But soon people tried to find out similar compounds as cocaine. They modified the structure of cocaine and were not successful because cocaine itself is very useful. It was only the modification of cocaine which led to success. And I will give you a few examples now for plant substances as models. Cocaine modification did not lead to success, but more than 1,000 substances were produced synthetically, and by chance aminobenzoic acid esters were found as potent local anesthetics, and acyl anylides, for example. I just took a few compounds out. They have not very much similarity to cocaine, but cocaine was more or less the starting material. The same is true for atropine and semi-synthetic derivatives, and here we can really find an improvement of nature, of natural substances. Atropine can be used in ophthalmology, for example, but has some disadvantages. So semi-synthetic derivatives have proven better suitable for therapy. Same is true for scopolamine and semi-synthetic derivatives, and you know all the antispasmodics derived from troponol alkaloids originally. And further on, synthetic anticholinergics were produced following the original structure of tropan alkaloids. Was it always improvement of nature which could be achieved? We come back to morphine, isolated 1805, as we showed, and it took 70 years until diacetyl morphine, heroin, was synthesized from morphine, and further on, a lot of substances, even until the 20th century, were produced with more or less better properties than the original morphine. Heroin, synthesized 1874, was used as medicine originally, and by trial and error, people realized that they came again from medicine to poison when the properties of heroin were detected, the danger of addiction and drug dependency. But this took some time. First it was used as medicine produced by Bayer until the other properties were recognized. On the other hand, there were several substances which improve properties derived from morphine by reduction, and a lot of synthetic analgesics were synthesized. We can find some improvement, although not all the expectations were fulfilled. Some of these substances have properties for drug addiction and drug dependency. Another example, cardiac glycosides. They were used as mixtures. You recall the trials of Dr. Withering, 1785, extracts with water or ethanol from Digitalis purpurea, later on Digitalis lanata, Scilla maritima, yielding the drug squill. And until the 1960s, such mixtures were used against heart insufficiency. But then people were successful in isolating the pure, single pure compounds, and the obvious advantage is that you can dose them very exactly in very small amounts. Again, the example of squill, Urginia maritima, is one of the names of the plants. In Vienna, we were successful in isolating more than 70 different cardiac glycosides from squill, and this had some consequences. Originally, the plant were named Scilla or Scilla. Then the name was changed to Urginia, and finally it was changed to Charybdis now, because botanists and taxonomists realized that it's not so simple. The systematics has to be revised, and there are several species of Urginia and many cardiac glycosides as mixtures in these drugs. We're coming to Albert Hofmann now. It's amazing that from the mixture, from this wild mixture of more than 70 cardiac glycosides, he was able, 1933 already, to get a pure crystalline product named silyrin A in the group of Stoll, and to find out the structure. And we must realize that at this time there was no NMR, no mass spectrometry, and no sophisticated methods of isolation either, or structure elucidation of substances. Prostylridine is used as a drug until now even. So Hofmann worked, as we heard in the morning, on cardiac glycosides, and only a few synthetic modifications are used until now from the pure substances out of cardiac glycosides with improved therapeutic properties. We come to the famous ergot, used by midwives already. We heard that in the morning again, but mainly it was known as a poisonous, and you know that many thousands of people were poisoned by ergot. And this is a famous picture of Matthias Grünewald at the altar of Isenheim, not far from here, and you find the picture of a human being poisoned by ergot. So ergot was well known as a poison during centuries, and you know all the morphology and the physiology of the plant, ergot, Claviceps purpurea, fungus, and this is the sclerosium on rye, and in springtime the sclerosia germinate, as we call it, and bring these fruit bodies, and so the cycle goes on. Now, from poison to medicine, you heard this morning already that the research performed at Sandoz in Basel was very successful. Stoll isolated ergotamine in 1918 already. Then the research was stopped at Sandoz, but when Hofmann was looking for a new field for research, he proposed to take up research on ergot alkaloids again. And you know all the successful story that numerous alkaloids were isolated and the substances were modified, the structure elucidated, and so on. Always using heroic organic chemistry without the modern sophisticated methods. And this is really amazing what Hofmann here achieved. Several products came into use as medicines, of course, hydrogen, dihydrogot, and so on. And also his problem child, why we are here, not only his birthday, but LSD. And this was discovered, as he himself in his modesty sometimes said, not only by chance, but also by perspicacity. That means by serendipity. He himself likes this term, which means that you find something which you have not been looking for. And it's quite unexpected and unforeseeable that you find something, and that's exactly what happened. It was planned research which he performed, but he found something by chance and by perspicacity. He was very interested to find out what happened to him, and this is perspicacity. Serendipity is the word he likes. So, is LSD medicine or poison? Was it improvement of nature? You will say yes. Other people said no, if you think of the United States and Washington. And obviously there are no simple answers to this question. This is Albert Hofmann, 1977, at the occasion of an excursion at the meeting of the Society for Medicinal Plants Research, the GA in Zurich, where he got the award of the honorary membership. It was 1977, and this is his wife here. Now I need not talk to you about the ergot alkaloids and other achievements we heard already in the morning, and this is taken out of his book, LSD, mein Sorgenkind, LSD, my problem child. Just one other example of more recent research. I show you a screening program performed by the NCI in the United States. There were more than 30,000 plant species and extracts thereof tested against anti-tumor activity. And those people were really lucky, because hadn't they had an extract of Taxus brevifolia in these 30,000 plant samples, they wouldn't have found any new drug. So you see, sometimes it may be very tedious and troublesome to find new active substances from nature. But they were lucky, and there was one new drug designed, Taxol, 1993. Here you see the structure of Taxol. It has the name Paclitaxel. Meanwhile, and there were several structure modifications. This shows attempts to improve the structure or the properties of Paclitaxel, and this was possible to some extent. And even in the needles, there are not large amounts, but some amounts of Taxol contained of Taxus baccata. Originally, Taxol was isolated from Taxus brevifolia, and this is the dew which you find at the Pacific coast in America, but you needed more than 3,000 trees to get 1 kilo of Taxol. So it was very important to find Taxol or some similar compounds that can be used and can be produced from the needles of Taxus baccata, which is a very common tree. Of course, there are many other examples for improvement of the therapeutic properties of natural substances. If you think of antibiotics, penicillins, for example, for oral use by modification of the natural substances, artemisinin for malaria treatment, silymarin, all the steroids which have been modified for several purposes, or tuberculin, and so on and so on. So finally, I come to mixtures from plants which are still used in modern phytotherapy. And it's amazing that these mixtures can be used as healing cocktails, and they have been used for centuries already without knowing anything about the chemistry of these extracts which were used. The identification of therapeutically valuable substances led to the standardized production of high-quality extracts today with reproducible therapeutic properties, and very common plant drugs can be mentioned here: chamomile, ginkgo, St. John's wort as one of my last examples. You know St. John's wort as an old medicinal plant, and an abundance of different molecules have been isolated from this plant, and the shape of the molecules is very different, so you can compare them with keys, and these keys fit to several locks to receptors in our organism, and the mixture is used against depression. The mixture is used. We have various different keys and locks within one plant extract, and I don't mention them all. Several receptors have been detected where St. John's wort extracts act on. We had clinical trials, and a lot of papers have been written on the antidepressant activity, and it can be used as a mixture. Probably lose other therapeutic properties if we take into account only the clinical trials. For example, the oil of St. John's wort is used for a long time as anti-inflammatory drug and for wound healing purposes. So if we follow the modern clinical trials, it's used only against depressions. We probably lose some other interesting properties of the natural compounds. And my last example is taken from our own work in Vienna. Yarrow, a very common medicinal plant, known for centuries. Achilles used the plant already for healing wounds, and there are several species of Achillea, and the water extracts are used, or alcoholic extracts are used as mixture, as healing cocktails. But if you look at the compounds which were isolated, just the group of sesquiterpenes isolated from Achillea, you see there are dozens of compounds which should be investigated in detail, chemically and, of course, pharmacologically. Can we improve the therapeutic properties of such healing cocktails? And if we can, how shall we do that? By isolating the pure compounds. This is successful. We know that from many examples from history. And we can use the pure substances, or even mixtures of pure substances could be used. Or, and that's what is done nowadays, preparing standardized products. We try to have medicinal products with reproducible quality, pharmaceutical quality and therapeutical quality, of course. How about mixtures from different plants? It's very complicated to find out the activities of these plants, but in several cases we have examples that the mixture of extras with thousands of active constituents are really useful as medicinal products. How can we remove poisonous substances? If you think of ginkgo, there are some substances which are undesired. So many questions if we use mixtures of plants, plant extracts, how can we improve the natural mixtures? So I come to a few concluding remarks. Evidently, the isolation of pure natural compounds is a story of success. Need not to mention that. Chemical modification in many cases, and we saw a few cases, not in all, leads to an improvement of nature, of the therapeutic qualities, the therapeutic properties, or even to interesting new qualities of activity, if you think of the atropine derivatives, for example. However, careful evaluation seems necessary, unavoidable, because we always have to be aware of unexpected, unforeseeable, undesired properties. They can probably only be revealed after trials with humans, as problematic they may be. Now, the vast majority of plants worldwide, approximately 500,000 of higher plants, are to be expected, and other organisms are not yet investigated. So a broad field is open for more investigations. And thousands of even known isolated compounds have not been evaluated pharmacologically. And there are an abundance, there is a lot of new test systems, and we will hear in the following 2 lectures about those. And known compounds have not been tested with new test systems. And we can be sure that an abundance of novel structures is to be detected. New leads for innovative drugs which can open up new areas of therapy. So altogether we have great expectations for the chemical improvement of nature. And I thank you very much for listening.
Friends, dear colleagues, dear cannabis experts, I recognize some here who did a lot of good research. Let me start first with psychedelics. The LSD is the topic of the day. In 1988, the Federal Office of Public Health gave me the job to synthesize LSD. Why and for what? Sandoz was not able or not willing to supply the Swiss Society for Psycholytic Therapy, LSD, for a big research project. So my job was to prepare 99% pure material. And this is a link to your subject. So I used ergotamine. I could buy this ergotamine. And by a simple modification of the substitution at the nitrogen, we finally got very pure LSD. Albert, greetings to Albert from far. He tried the material and said, this is by far the best LSD I ever had in my hand. This is not PR for me. And we were working very carefully with mouse protection, gloves, everything. I'm not a better chemist than Albert, but we knew exactly what could happen if you are not working carefully. So any one of us, anybody in my lab, the technicians say, really experienced the pharmacology of LSD. But a couple of years later on, I had the chance to participate in a psilocybin study in the psychiatry department of Zurich. 20 years, I told my students what psilocybin is, how it works, from the school book. And from then on, I really realized what psilocybin is. I got a very high dosage, 20 milligrams, which is really at the upper limit. This would really do not fit in this frame of my talk, but just to start my experiences in these fields, and I can say it changed a little bit my life. Anyway, this was the only time I made this experience, but it was very impressive. Okay, let's go to the topic of cannabis. A colleague in the States once said, you can read it here, I don't read you. And I think this really says that we don't have an and or, so we have an and situation, exactly. And for me, this plant is really one of the most fascinating plants I've ever worked on regarding politics, regarding molecular biology, regarding botany, chemistry, and so on. You see, more than 25,000 papers have been published. So besides Nicotiana tabacum, I would say it's the most well described scientifically, best described plant in the world so far. Let's start in old China. The use of cannabis as a medicament goes back to Shen Nung, the Chinese emperor, about 3000 before Christ. So he already described the therapeutic potential of cannabis. And then this famous lady, Maria Treben, she wrote, and I decided to give you this text in German. I come back to that aspect. When you see here headache and the difference between empty brain and the pathological situation, she was already able to differentiate what happens under normal conditions and under pathological conditions. And the last sentence especially, I would like to point on. You see, she already had some ideas about toxicity of cannabis. Then, in the late 80s, what was supposed for many years in relation also to the endo-opiate system, the endorphin system, a group in the United States discovered the first, I'm sorry, I go back, first discovered the Cbr One Receptor. It has been fully elucidated in its structure. You see it has 7 transmembrane structure and is G protein coupled. I don't want to go into details. And what is very interesting is that this body system of cannabinoids is much older than this endogenous system, than the exogenous, the plant cannabinoid system. So it's maybe 10 times more, it's an estimation, maybe 10 times more old than the body cannabinoid system. We can only speculate why this is so. Maybe you have some explanations. And you see that this CB1 and endocannabinoid system is not only occurring in mammals, but also in other creatures of the nature, like in the Blutegel, leeches, birds, amphibians, sea urchins, mussels, and so on, but not in insects. Why not? We don't know. When there is a receptor there must be a ligand. So in 1992, the first ligand has been identified by a group in Israel. This ligand has been named arachidonyl ethanolamide, and with the trivial name anandamide. Ananda from Indian Sanskrit, which means bless, and amide, this is the chemical term for the structure. And then later on came another receptor type, the CB2 receptor. It has been found in rat. Then in 1994, the big pharmaceutical company, Sanofi Aventis today, they synthesized in the lab the antagonist, a tool, a most significant tool for pharmacological research. Today we have a medicament based on this compound, which is called rimonabant. Acomplia will be the name on the market. It's not yet on the market here in this country, but it will come soon, as I heard. I come back to this very interesting compound. Has chemically nothing to do with cannabis, but it acts at the same system where also cannabis is acting in the body. 97 other ligands were identified, and there is really convincing evidence that in near future another system will be identified. The CB3 system. Just to show you how complex the situation is. Everywhere, almost everywhere in the body, you have receptors for cannabinoids. Starting, of course, in the brain. CB1 is mainly located in the brain. CB2 mainly located outside of the brain. Gastrointestinal tract, reproduction organs, bones, nerve terminals, bladder, etc., etc. I think, Dave Pate, you agree that much of what you have described can be explained by the presence of CB receptors in the brain, in the eye. I come back to this aspect later on. Just to make the situation more complicated, and it's really difficult to understand what is going on, you see all this very important neurotransmitter and other physiologically very important system in our body is linked in a certain respect with CB1 and CB2. So CB1 is activated by plant cannabinoids, and of course with our body cannabinoids. And to complicate even more, there is an interaction between these two cannabinoid types, the cannabinoids from the plant and the cannabinoids from our body. I once have been asked, why do we not experience a cannabis flash and cannabis psychotropic effects every day, because we have cannabinoids in our body? I must say, anandamide, the short life of anandamide is really very short, so it is synthesized and then it is metabolized very quickly, which makes the pharmacological research not easy, I tell you. Physical stress. What has physical stress to do with cannabis or cannabinoids? It has been shown on students in California that the endocannabinoid and the endorphin levels are increased under physical stress. They did the experiment on home trainers, on bicycles. But in addition, and this is new, also the endocannabinoid system is activated under physical stress. And there is a link between these two systems. So can we say that the jogger's high? And I don't know who is jogging here. Euphoria of joggers is well known. Also, is this really due to endocannabinoids and or endorphins? But there is evidence really that the endocannabinoid system is activated. And you can measure that in plasma. The level is really increased. So something is happening. What is the physiological role of the endocannabinoid system? What we know today is that basically the endogenous cannabinoid, anandamide, the first one, has similar effects than THC, but the effect is shorter and less intensive. This has been shown in animal studies. It has something to do with the tuning, fine-tuning of neurons in brain areas which have something to do with memory and movement coordination. Pain, feeling, sleep. I come back to sleep quality under influence of cannabis. Appetite, a very important aspect concerning the therapeutic value of cannabis. Reproduction, feeding behavior, then gastrointestinal functions, inflammations, and so on and so on. This list is not complete. A recent patient survey concerning the use of cannabis in the folk medicine in Germany, Switzerland, and Austria showed the following things. These were the diseases mostly mentioned. Other diseases, what is interesting is back pain, hepatitis C, sleep disorders, epilepsy, spasticity, headache, migraine, very often migraine, appetite loss, the Tourette syndrome, the tic syndrome. A clinical study has already been finished which showed really the very positive effect of THC in Tourette syndrome patients. It has been performed in Germany. And one question was, why are you using cannabis? Of course, to alleviate symptoms, of course, to be sedated, to get a little bit of euphoria, antidepressant effect, to reduce anxiety, and so on. These charming elderly women, when you look very carefully, you see something here. They wear little bags full of cannabis. They are not smoking cannabis, of course. They are not used to the effect of cannabis, and this is another generation, of course. So why? They say, and they really claim, they can get rid of all the school medicine, sleeping pills, and instead of just wearing these bags with cannabis, it increases sleep quality dramatically. A controlled clinical study has not yet been performed, so these are anecdotal reports, of course. But anyway, I think it's quite interesting. What's the active principle? Not THC. THC is not volatile, of course. They are not smoking. It's just hanging here. But they smell the terpenes. They know about the effects from folk medicine. So is it a placebo, a pure placebo effect? We don't know. Anyway, school medicine does not believe in this effect. And until we have real convincing clinical data, but these are not occurring so far. A very promising form of application is the sublingual spray. It has been produced by GW Pharmaceuticals in UK. Bayer bought for, I guess, 50 million euros the right to sell the product. It appeared on the market recently in Canada, and the company is waiting for approval in EU, and maybe we will get the product too in Switzerland. We will see. What is new? This is a standardized cannabis extract. This is real nature. It's not a synthesized compound. The plant material has been produced under GAP conditions, meaning really state-of-the-art conditions, by cloning. The production facilities, of course, are not known in public. They are somewhere in UK. And indications are so far spasticity, chronic pain, neuropathic pain, and cancer pain. Every person who has the experience of a joint knows his appetite is increased. He goes to the refrigerator and eats something, or eats a chocolate or any other food. So this is the result of CB1 agonism. We can use this experience to increase the appetite in patients who suffer from a dramatic loss of appetite, AIDS patients, cancer patients. And this is, in fact, really done. And this indication is also approved by FDA in a medicament, in a drug called Medicinal, which is synthetic THC. Has nothing to do with cannabis, but it is synthetic THC. So this problem is also very well known. Can we use this effect? Of course not, but we can use this principle by blocking Cb1 receptor, which has been proposed by Sanofi. So this new medicament, rimonabant Acomplia, blocks Cb1 and is proposed as an anti-obesity drug in the metabolic syndrome. And hopefully the result will be like this. And you can imagine how enormous the market will be in the future, only in the States, but also in this country. We know that 30 to 40% of Swiss population has overweight. Really, the market is absolutely enormous. During the clinical studies, they realized that people who were smoking patients suddenly stopped smoking. So there must be a link in the brain between the appetite controlling center and the abuse structures in the center for nicotine, the brain rewarding system. And even here, the market is absolutely enormous, of course. And we think that the indication smoking cessation will also be approved by the regulatory authorities. Very interesting experiments from animals showing before and after administration of THC, quite a high amount, but anyway, it has to be discussed. And you see this very aggressive form of a brain cancer, how the size is reduced very quickly. And these first experiments show that cancer, what kind of cancer is still open, but maybe brain cancer, but also other forms of cancer, can be a very interesting field for therapeutic use of cannabis or cannabinoids, of course. migraine. Coming back to Maria Treben. Molecular biology, don't look on the details. It's quite a complex situation. But what is interesting is the Cb1 receptors are located very closely to the system which is responsible for the development of migraine. So we have here the CGRP, calcitonin gene-related peptide. This is a key player in the pathophysiology of migraine. And very close to this system, we have the Cb1 receptors. And when we administer endocannabinoids or exocannabinoids, phytocannabinoids, the result is a blocking of the CGRP system. And it has been shown, and we know that from folk medicine. I know migraine patients who say the best medicament to treat my acute migraine attack is a joint. Of course, this is pharmaceutically not acceptable. Meaning it's our job to find alternative galenic forms, application forms, which is just going on. We try to develop an inhalation form for cannabinoids. Not by smoking, of course. Epilepsy, just to come soon to the end. Very well known from folk medicine. And what is interesting is here a study going on in Italy in children who are therapy resistant. And CBD, a non-psychoactive constituent of cannabis, reduced significantly the number of seizures and the intensity of seizures. Also very promising new therapeutic field for cannabinoids. ALS, amyotrophic lateral sclerosis, a study going on in Switzerland, in St. Gallen, at the Cantonal Hospital of St. Gallen. We are doing there in cooperation with the ALS clinicians. A big problem in ALS is spasticity. This is really reducing life quality dramatically. So we use here the very well-known effect of THC reducing spasticity in multiple sclerosis patients, or here in brain-injured patients, or here in ALS patients. This is Mrs. Musica from Florida. She was visiting us in Switzerland. She got the permit from Bill Clinton to use joints to treat her eye disease successfully. It was very astonishing. She was fighting, she told me, 12 years to get that permit from US government with her physician, and today she is only smoking cannabis cigarettes. Of course, this is not an ideal situation, but on the other hand, she would be blind today without cannabis. She claims, she predicts. Do we have a cannabis, a plant, or a non-cannabinoid or a cannabinoid situation? We have on the left side the plant product. So it's something like a shotgun therapeutic concept. On the right side, we have the monosubstance situation, which is more a silver bullet situation. Both are leading and ending in the CB, in the endocannabinoid system, influencing transport mechanism, synthesis, metabolism, and so on. Hopefully we have a therapeutic effect, meaning and this is the conclusion, we have also here an and not an or situation. We have an and situation. I hope that cannabis will have a future, that we have this left arm here and not only the right arm here. We will see what happens. And to conclude, the therapeutic potential is summarized here, as known so far. I'm convinced that maybe in 5 years we have more parts of the leaf, but we'll see. We cannot forget this side effect. Sometimes this side effect can be a main effect. We did a study here in Basel on brain-injured patients. Paralyzed patients are usually also depressed, and the cannabis side effect was very useful to reduce sometimes the severe depression. So the clinicians accepted the side effect as a main effect. So will this be the future or not? We have still a discrepancy between empirical-based data and evidence-based data, of course. There is a lot to do. Every project is expensive, every clinical project. However, 15 years now of endocannabinoid research has already opened the way to new and very promising treatment strategies. And this is my message I really want to leave here in the audience. And Albert is not here. When I was visiting him the last time, last fall, he was in excellent conditions, and I would like to finish with an experience I had with him, not an LSD experience. But as a young assistant, I was presenting a poster about CAT, the African stimulating drug, at the Society of Medicinal Plant Research. So Albert came to my poster, was looking at the poster, said, yes, very interesting. We described the new active principle. And then he asked, did you try that material? So I would like to finish with that.
Thank you very much for the kind words of introduction. I'm very pleased to be here in this very memorable symposium, and I think it's a rather unique situation to speak at such a meeting that has kind of a global impact, actually, if you look at the media and the interest in the field, not only of LSD, but obviously the source, actually, that is at the core of this discovery, natural products. Well, in the two preceding speakers, and in particular the first talk has addressed actually the historical developments of natural products in medicine and made the bridge to the current use as medicines. And what I'm going to do is actually look into the present situation and trying to give you an outlook in the future of natural products, and the way how I personally perceive that natural product research is to go in the future. But let me start with some maybe rather basic issues first. What are we looking for when we talk about drug discovery? We are looking for medicines. And now what are medicines for? To address a disease. And if we look at it from a biochemical aspect at disease situations, then we are dealing with biological, biochemical processes that are somehow dysregulated for various reasons. And typically now in a cell, the essential molecules, the class of molecules that are involved in health and disease are proteins, macromolecules, and typically drugs would target actually these proteins, these molecules, and what we want then to do is in some way to modulate specific proteins in their function as to address this dysfunction that leads to the disease. And so we try to find molecules that specifically normalize this regulation in some way. So this is a very simple, obviously rather simplified explanation, but just one example. Oh, sorry, that was a mistake. Just an example here, protein kinases. These are enzymes that phosphorylate, introduce a phosphorus group into proteins, and this is the fundamental principle of activating proteins in their function. So these protein kinases activate other proteins, activate biological processes. And if this is dysregulated, for example, too much of these activating processes, this leads to diseases, for example, like cancer. And so inhibitors, for example, of such protein kinases then normalize this overactivity and inhibit cancer tumor growth. This just as an example. Now, the molecules that we are looking for, they are represented here as small molecules in the skeleton, and what you see as a shade in the back is actually the macromolecule, the protein. These small molecules, typically that we are looking at, they have to interact with this macromolecule in a way as that the function is modulated in the desirable way. Now, they bind to a certain specific position of this molecule, and there are different ways how they can modulate. They can inhibit the catalytic activity of an enzyme. They can block a channel that leads, for example, ions from the outside of a cell into a cell. Or we can also have the situation that the binding leads to a change in the conformation, the shape of the protein, and activates the protein. Or we can also have the situation that they inhibit or change the interaction between proteins in the cell, which is a very important process as well. Now, we have so far been looking only at single proteins. Now, the situation in the cell is obviously much more complicated. Biological reality is highly complex, and the situation is rather like this. It's like a traffic jam somewhere in a major city. So we have a complex situation, a highly crowded situation, and this is the reason why all these processes have been highly regulated. And these biological molecules that are here just shown in that case in a bacterial cell, there are different kinds of proteins and other molecules. They act in part independently, but also in a major part in very complex networks. And now somehow here in this kind of messy situation, the drug should act hopefully rather selectively. Now, I just want to make nonetheless a quick look back in the history of drug development, and actually just want to show you what the impact actually has been of natural products in our current pharmacopoeia, in our current drugs. And if you look at the inspiration or the sources for the current drugs, and if you look at them from the different origin, then you will realize that biological sources, plants, animals, including humans, microorganisms, have been the major contributor to our drugs in a direct but also indirect way. But if we look maybe more to the recent past, this chart here, this pie chart, shows you the origin actually of the new small molecules, new drugs, new chemical entities, NCEs is new chemical entities that have been introduced in the 2 decades from 1981 to 2002. Now, if we have here 39% synthetics, and the rest is all directly or indirectly inspired from natural products. Only a small portion direct natural products used as drugs, but many natural product derivatives and compounds that have been inspired by natural products. So the impact is still very high. Now the question, why have natural products been so successful in drug development? And I want to try to give you maybe a certain part of the answer with this slide. Now, on this slide, you see 8 compounds, 4 natural products here on the top, and 4 synthetic drugs, well-known drugs here. These are also mostly here also drugs. And now you compare these compounds. There are major differences between natural products and synthetic drugs. And the major point is the structural complexity first, and the fact that these natural products have many so-called chirality centers. That means these structures, which are shown here in the planar projection, in fact, they are 3-dimensional. They have a complex 3-dimensional structure. And this is much more complex than these compounds, which are kind of more boring in their 3-dimensional structure. And why is that so? That's, I think, the major issue. Natural products are products of biological selection processes. They have some reason to be there. And they have been kind of designed in their evolution to interact with biological macromolecules, meaning proteins. Why? During their biosynthesis, they interact constantly with various enzymes, which are obviously proteins. And also, as we know, many of the targets of these compounds are proteins. So they have kind of 2 reasons to be designed to interact. And this means knowing the conservative situation for many biological targets relevant for medicines, that these compounds, which have been evolving not to serve for us as drugs, but that they address some target in an animal maybe, or in a microorganism, they can be useful for us. Now, we have already heard several times this morning about some of the plants shown here. Traditionally, the early drug discovery and isolation of natural products started usually from traditional knowledge, empirical knowledge of the activity, of the toxicity of certain plants, or in that case, actually, it's a fungus. And very important molecules came out of that are still in use. And you have heard actually about these 2 plant products here, digoxin and ergotamine, which Dr. Hofmann has been involved with during his research at Sandoz. Now, that was the situation in the past. Today, the tools that are available in the pharmaceutical industry for drug discovery is much more complex and can be maybe kind of summarized in this picture. So we see somewhere natural products here in this corner, but you see all the rest around here. So there are many different strategies that are being used. For example, synthetic chemistry, high-throughput chemistry, combinatorial chemistry, but also theoretical approaches using computing and many other approaches, which then all lead to a so-called hit, which leads then to lead compounds, lead structures, which are the kind of the first step in the development of a new drug. So you see only one corner here in this whole picture are natural products. I don't want to discuss whether it's meaningful to kind of reduce this natural product research, But I just want to show you maybe now looking into the more into the future, actually starting from the present, where natural products can be of importance and how actually new technologies are shaping and changing the way how we can actually do research on natural products. And I would say there are basically maybe four, in my opinion, four major aspects that are driving present and future natural product research. And I've summarized them here: miniaturization and automation. And this is driven by certain technologies like modern analytical tools, and miniaturization of bioassays, which allow us, as you will see, the identification of active natural products with sub-milligram amount of material. The enhancement of structural diversity via addressing new pools of our biodiversity. And this means access to unexplored ecosystems and new approaches in taxonomy. There's another point just here in silico, computational methods that can be used that, as we will see, allow us to do screening just on the computer for looking for new drug leads. And finally, I will then end with a very recent aspect, namely moving to a new paradigm in biological, in the biomedical research, moving from single to aspects to global assessment of activity, which is driven by the so-called systems biology approaches, and which allow us, and that's the most important point, to move from a hypothesis-driven research towards an essentially unbiased approach in understanding the activity and the function of molecules. Let me start with the first aspect first, miniaturization automation. How can we discover in the extract, and you have seen how complex these extracts are, they contain hundreds of compounds, how can we find an active entity in this bewildering mixture? Today, we can use an approach that is called Hplc-based activity profiling. We separate milligram amounts of extracts on special devices, separation columns, from which individual compounds then elute in a solvent one after the other. So each peak here represents ideally a compound. There are obviously many more. And we can, from these amounts directly by certain detectors, obtain structural information which is very complex. And on the other hand, we can directly go to a bioassay and find activity. And then we can make the correlate between bioactivity, a peak here, and a structure. And this is possible to do it with microgram amount of extract and with milligram amount of extract and microgram amount of pure compound. And obviously with certain technologies. That's just shown here. This is a typical equipment that one uses for this purpose. Here we inject the sample, then it's separated in here. We have different detectors, and we can fractionate here into certain devices for the bioassay. And the bioassays, as you can see here, these are so-called microtiter plates, which have about this size, 10 to 15 centimeters, with many small wells. And in these, we carry out bioassays in volumes of 200 microliter down to sub-microliter volumes. And just a practical example that I want to show you that it works in reality. This is from our own research. We were looking for the anti-inflammatory principle in a traditionally used medicinal and plant, and the dye plant, Isatis tinctoria, the woad plant. And so here you see actually this separation, and you see different peaks here from the separation of the extract. And above, we have the activity, cyclooxygenase. This is a key enzyme in inflammation, the inhibition in these different fractions. And we see here very strong inhibition here, but if we expand that, we have many more peaks, obviously. And ultimately, it was possible to pinpoint this small shoulder as being the active compound with this structure, a highly active molecule. And all it needed was 200 microgram of extract, and for the bioassay, we only needed, for all this, 50 microgram of material. The second point, biodiversity, accessing new sources of natural products. What traditionally we have been looking at have been obviously the biodiversity that we can see in our macroscopic world. So what you see here when you go out in the woods, what do we see? We see basically mostly higher plants. But the biodiversity, actually, the true diversity is hidden. This is not the macroorganisms. These are microorganisms, fungi, bacteria, and there are the other ecosystems that we do not see, like, for example, marine systems and so on. Just to give you some figures where we stand. The plants, obviously, which have been the most prolific source in the past for finding new medicines, as you have heard, we know most of the species, 260,000, these is always in thousands. There are some more around, but we basically know all the plants. They've been quite well investigated. But if you look, for example, at the fungi, we know only estimate 5%. If you go to the bacteria, less than 0.5%. And these are big numbers, 1 million, 1.5 million of estimated species. And if you look down here in the animal kingdom, then you see here that the insects are probably the most successful group of organisms anywhere in the history of the world. 8 million estimated species, 12%. So there's much more to look at for new natural products. And just to show you two examples of compounds that are now in clinical trial and very likely to go into the introduction as clinical drugs, just from some rare sources. Here, the epotylons, which are produced by the so-called myxobacteria. These are gliding bacteria, a very unique group of microorganisms. And these compounds, they inhibit cell division by inhibiting the disassembly of the microtubule system, and compounds are now in advanced clinical trials. And if you look into the marine system, there are many marine microorganisms and algae and other lower organisms, but interestingly also fish can even give some useful compounds. This steroidal compound comes from the shark liver, and this is again a compound that is in clinical trial as an anti-cancer drug. What does the compound do? It inhibits the vascularization of tumors. Now let's move to the next aspect, in silico screening. Structural biology has made tremendous advances in the understanding of the protein structure and the interaction, for example, with their natural ligands, for example, neurotransmitters, etc. And this comes mostly from the fact that, that it has been possible to crystallize certain proteins and to make X-ray crystallography of these compounds. And with this understanding, it is possible to draw their three-dimensional structure, and then one can then do virtual screening on the computer for molecules that bind with the active site, for example, of a protein, like an enzyme. And just to show you one example here, we are looking here at acetylcholinesterase. Acetylcholine is an important neurotransmitter which needs to be broken down very quickly, and but on the other hand, acetylcholinesterase, if we inhibit this, this can be useful therapeutically in diseases like in Alzheimer's disease. And so one can co-crystallize this protein with some inhibitors like this one, this galantamine, which is a natural product from Galanthus nivalis, the snowdrop. And this allows to show then where the interaction are between the protein and the inhibitor. For example, here, proton donor and acceptor functions interactions which are of hydrophobic nature. And what we see in the back, in this backbone, is actually the cavity of the catalytic site where the enzyme cleaves the neurotransmitter. So from this understanding, one can then draw pharmacophore models. That's kind of a special model representing the interactions. And this is shown here. So these are the spheres of interaction, and behind is kind of the shape constraint due to the cavity of this active site. And then one can use this model to screen natural products, just without having them in hand, just on the computer. And just an outcome of a screening that was done recently, not in our lab, in another group in Austria. They screened 100,000 natural products in this model, and they found some inhibitors. Interestingly, in particular, this compound here, scopolamine, which fits into this model very nicely, and interestingly, not only binds in silico, it also inhibits the enzyme in vitro. So this is an interesting approach. It has some advantages. You don't need physically a compound in hand. You don't need an enzyme in hand. You can just do it on a computer. The limitation, obviously, is only possible for proteins that have, where the shape, the structure is known, and it's only possible to screen known compounds. So this is obviously a limitation. And the other point, and this leads me to the last aspect, is these approaches are all very reductionistic in their approach. We look at the single molecule interacting with a single enzyme. And obviously, biological reality, nature, is much more complex. So the global assessment of activity, that's the last point with which I will conclude now, is actually moving just from this reductionistic approach to a global approach. Because if you look at the cell, if you look at a tissue, at an organ, pathological processes do not only involve a single protein, but the entirety of the cell is somehow involved. And now we need to understand the complexity of this reality. This is the approaches that are being used now since recent times in systems-oriented biology, where one tries to understand the entirety of the information in a cell, looking at the protein level, at the metabolites they also produce, also looking at the genetic level. And putting all this information together allows us to draw some patterns, some conclusions, and to use these patterns to generate some hypotheses, for example, for disease, but also to test drugs in their response. And I just want to show you one aspect, namely, when we move from the gene to the protein, we need this transcription, which is kind of done by messenger RNA. And so this step, for example, can be looked at in a global manner, but also the other aspects which I will not talk about. How is this done? This is done in miniaturized devices like this, the glass slide, which has thousands of cells which contain a certain piece of DNA which is complementary to the RNA which is produced during the transcription from the gene to produce the protein. And so one can actually, from this device, now draw conclusions. We lyse the cell, we transcribe this RNA with a fluorescent tag and bind it to this surface. And then we see the interaction due to the complementarity of this RNA to the cDNA, and the intensity of the fluorescence due to the tag reflects then the state of the expression of a certain gene. Because not all genes are active at the same time, and the activity pattern are very important. That's how it looks like. A human genome array with 30,000 genes. So we can see pattern of regulated genes. We can from that then identify the protein, the gene product that would be produced by that upregulated gene. One can put that in a functional context and draw hypotheses from that. And I'll just show you what it looks then in reality. We have been doing such approaches, for example, for studying phytomedicines and also pure natural products. Maybe some of you may be familiar with this plant, black cohosh, which is increasingly popular as a phytomedicine for the symptomatic treatment of postmenopausal disorders instead of estrogen replacement therapy. This is very nice, but the problem is one doesn't really know how this black cohosh acts. There's been hypotheses, phytoestrogen activity, selective estrogen receptor modulators, other mechanisms. Now, since estrogens act on nuclear receptors which act on transcription activity, an approach like using an expression profile is an ideal approach. And just to show you what outcome can be when we take such a black cohosh extract and test it in a genome-wide expression profiling, 480 genes out of these were regulated up or down-regulated to a certain, let's say, significant extent. One can then group these genes according to their functional affiliations, cell proliferation, growth, signaling, etc. And then one can put actually these gene products in their function in functional networks, which look highly confusing. This is just the example for the effects on the cell cycle. And if you look at the cell cycle progression here, we see that we have many genes here downregulated here in this phase, moving from G1 to S phase in the cell cycle. And so we have an anti-proliferative effect, which we can see in the cell culture, but also we can hypothesize that the action is taking place here. The advantage here is that we can have we can test unknown compounds for new functions, we can test complex samples, and we can generate new hypotheses and look at the complexity of biology. So where do we stand today with natural product research? We have many compounds, more than 200,000 are known, but we know we have limited knowledge of our biodiversity, as you have seen. There are certainly hundreds of thousands of more compounds around. Few compounds of these have been really investigated for biological or pharmacological activity, and also we have a limited understanding of the function of secondary metabolites for the producing organisms. So in a way, we even don't see the tip of the iceberg, to put it in a picture. On the positive side, we have a growing understanding of the evolutive processes in natural product biosynthesis. And this understanding helps actually understanding the function and also gives us growing insight into the molecular mode of action. And we have, as you have seen at the end, new tools for global approaches for looking at natural products. So I would again want to put it in a picture, which is this one here. So we are in the state that we can see new horizons. We are still very limited, but I think the future for natural product research looks, in that sense, thanks to these tools, quite interesting. Now, if we look at this bewildering biodiversity, these incredible differences in shapes and ecosystems that these organisms live in, I think there's still plenty to do, and there's probably some molecule for some disease, for any disease, somewhere. The question is, how can we find these molecules, and how can we intelligently test these molecules for their function? And I think that's the challenge that we are facing, and the best what we can do is make a good use of these resources. I thank you for your attention.